in order to address this problem of antibiotic resistance. And many of us may or may not be aware of this, but it is among the top 10 public global health threats that is facing humanity. And this resistance is often attributed to the fact that we misuse and overuse our antibiotics. And a really nice example of this, or should I say not so nice example of this, is during the COVID-19 pandemic, where the United States saw a 15% increase in infections that were attributed to drug-resistant pathogens. And this is really gonna start complicating our ability to treat common infections as well as perform life-saving surgeries. So if we don't nip this problem in the bud, we're gonna have some serious implications in the future that we're already suffering. So with that, there's an urgent need for alternative therapeutic treatment strategies. And the historical approach has been to target or to kill bacteria by targeting essential processes like those I've depicted here, from membrane integrity all the way down to protein synthesis. And bacteria, in order to cause an illness, have these tools known as virulence mechanisms. And our approach is to disarm bacteria by targeting those tools, essentially. So I've depicted a small subset of these virulence mechanisms, and these can be anything from invasion into our cells or evasion, as well as resistance to just general antibiotics and our native host immune systems. So with this strategy, we are not only neutralizing the bacteria, but we are also trying to preserve bacterial viability. And this has the added benefit of minimizing the selective pressure to evolve resistance. So you may be wondering, how exactly can we target all of these tools considering there are so many? Well, we leverage two-component systems. And these two-component systems are key signaling pathways that are heavily implicated in the regulation of these virulence mechanisms. So let's take a closer look at what exactly these two-component systems are. Well, they're comprised of this blue sensor histidine kinase and a response regulator depicted in pink. And this response regulator typically acts as a transcription factor. This histidine kinase has three main components: this signaling domain, this dimerization histidine phosphorylation domain that contains a catalytic histidine, and a catalytic ATP binding domain. And these systems are responsible for responding to environmental changes. For example, when a signal is sensed, it'll bind to the signaling domain, prompting ATP that is bound at the catalytic domain to undergo dephosphorylation with this histidine residue. A phosphoryl group transferred to the response regulator will elicit some type of cellular response, similar to those virulence mechanisms that we were talking about. And what's particularly attractive about these systems as therapeutic targets is that they're ubiquitous among bacteria. Oftentimes, a single cell can have upwards from 10 to 100 two-component systems at a time. They're also absent in mammals, which allows us to selectively target these bacterial cells. And moreover, they are non-essential. So this allows us to preserve the bacterial viability. And finally, and most important to my work, is the high degree of conservation in the catalytic domain. We leverage this conservation to inhibit those virulence mechanisms through antivirulence inhibitors. So taking a look at previous work in our group, we conducted a high throughput screen that yielded several analogs that had this conserved 2-amino-benzdiazole motif. So let's take a look at those now. And something that was really interesting about this was that this R1 analog that has a trifluoromethoxy group is actually a treatment for the Lou Gehrig's disease, which was very promising to us to know that this scaffold already had some type of therapeutic associated to it. So taking a look at their ability to inhibit activity of that model, of our model histidine kinase, this IC50 is just a measure of potency for these analogs. And what we can see here is that several of these analogs had potency ranges in the single micromolar range, which is exactly what we wanted them to be. Transitioning these into an antivirulence assay, specifically, we decided to assess their ability to inhibit motility of a pathogen. So this assay works by utilizing a soft agar, and within that agar, we can dose our compounds. We can then go ahead and inoculate a small subset of cells and measure the changes in the growth and compare it to the untreated control. And here what I'm showing you is that this dimer analog actually had a 92% reduction in that motility. So that was really promising. And that encouraged us to proceed further with another structure-activity relationship, with the goal of identifying motifs that were responsible for the activity that we were seeing in order to generate better inhibitors later on in the future. So here are our most promising subsets from the structure-activity relationship. And once again, we looked at their ability to inhibit that HK activity. And again, we have potencies in the single micromolar range, which is great. But transitioning them into that motility assay we talked about, this is where things start, we start seeing that discrepancy. Specifically, if we take a look at the C38 analog, it was very potent in vitro. However, when we transitioned it into an in vivo study, it had no inhibition of the motility phenotype. So with this, unfortunately, we were unable to yield any clear trends from the structure-activity relationship. And this is where my work comes in. The goal of my work was to develop an affinity-based probe to determine the cellular targets of these non-covalent inhibitors for the rational design of future inhibitors. So let's take a look at what an affinity-based probe is composed of. It's a tripartite structure that contains a directing group. In our case, this 2-amino-benzene thiazole scaffold. This allows us to guide the compounds to their intended targets. We then have a reactive group. There are several types of reactive groups, but this allows us to covalently modify amino acids that are within the binding site of these compounds. And we specifically were interested in this electrophilic sulfonyl fluoride, considering its range of reactivity with various amino acids of nucleophilicity. And this was specifically important for us because we lack the traditional cysteine amino acids in the catalytic domain. So this would allow me to, react with a lot of different amino acids. And finally, we have this click handle. And this click handle allows me to append fluorophores or affinity tags such as biotin through click chemistry. So the first question with this probe design in mind was, is this covalent reactivity similar or analogous to our non-covalent inhibitors? We needed to answer this question first. For example, did it inhibit that HK inhibition the way our non-covalent analogs did? And did they have that anti-virulence activity? So I started off this work by generating a small library of covalent inhibitors with the objective to determine the proper placement of the sulfonyl fluoride on this arrow ring relative to the heteroatom linker. So one of the first things I needed to overcome was that the meta and the para starting materials were commercially available. However, the ortho starting material was going for 500 megs for about $800. And I didn't want to make that commitment quite just yet. So I started from some things that I could work with in lab, and I developed this synthetic route. Starting from 2-thiocresol, I went ahead and did an oxidative chlorination to install this sulfonyl fluoride. From there, I furnished the sulfonyl fluoride using KF, and brominated that methyl group using NDS, and afforded the starting material in pretty reasonable yield. So with all of my starting materials in hand, I can now start developing that library. And I'm only showing you start. So typically, all of these products underwent just some type of nucleophilic substitution of an aryl bromide or an aryl acid chloride. And here are some of the derivatives that all of the derivatives that I synthesized. But something really interesting that I observed was, even though I put in all of this work to make the starting material, that ortho analog was essentially not really useful to us. During purification, I actually observed this intramolecular cyclization with the proximal amine to form this sultan derivative. So right off the bat, that told me that the ortho analogs were probably not going to work, and I could, I'm not really sure what to do with that. So from here, taking the rest of the compounds that I could use, I started asking myself, do these covalent analogs inhibit the activity of our model histidine kinase? So let me take you through what that assay looks like. It has two main components, this HK53 protein, and what's important about this guy is that it is constitutively active, which allows me to bypass the need to input some type of signal to activate it. It also possesses the conservation in the catalytic domain, which is exactly what our compounds are targeting. And then we have this Bodibi ATB probe. And this guy will only label active histidine kinases. So for example, when this catalytic domain is accessible, this probe will bind and transfer this thiophosphate Bodibi group to covalently label this histidine kinase. When I load these samples on a gel, I can actually detect the fluorescence of this Bodibi analog. However, when I pre-incubate this protein, well, let's just say an inhibitor, well, this inhibitor is gonna occupy that catalytic domain. This will no longer allow the probe to bind, and therefore we can see a decrease in that fluorescent signal. So walking through this data really quickly, as I'm increasing the concentration of that inhibitor, I'm occupying more of this domain and allowing this probe to essentially label this catalytic histidine residue. So we're seeing that decrease in signal. So working up this gel data, I could generate a dose response curve. And from this dose response curve, I can calculate this IC50 parameter. So this IC50 parameter is a measure of potency of a compound at inhibiting 50% of a specific biological activity. And in our case, it was at HK inhibition. So looking at the calculated IC50s, we can see that all of them had potencies in the single micromolar range, which was really good, right? But more importantly, and that was very similar to our R1's non-covalent inhibitors. But more importantly, this assay allowed me to determine that these inhibitors were binding to the intended target, the catalytic domain. So from here, I wanted to determine if these covalent analogs were affecting viability. As I previously told you, we don't want these analogs to affect bacterial viability. So to do that, I leveraged bacterial growth curves. And these growth curves allow me to treat bacteria and monitor their growth over time. So what I'm showing you here is treatment of this Salmonella enterica strain with the covalent inhibitors. And what we can see here is that the analogs 1B and 1C actually mirror the DMSO really nicely. And this was indicating to us that these were not having any issues with viability. However, the same can be said about 2A and 2B, where we can see this reduction in the final optical density. And this was indicating that these analogs were having some type of effect on bacterial viability. So from here, the last question that I needed to ask myself was, do these compounds possess antivirulence activity? Specifically, can they inhibit resistance to antibiotics in resistant cells? So previous work in our group utilized this R1 scaffold and found that it could actually enhance polymyxin E antibiotic activity in resistant cells. So I utilized a checkerboard assay. This assay tests combinations of an antibiotic and an inhibitor in order to determine if the combination of treatments works better than the individual treatments themselves. So looking at this table, polymyxin E starts killing bacteria at approximately 64 mg per ml. When dosed with our R1 compound, we can actually decrease this to 8 mg per ml. So taking a look at these covalent analogs, two of these guys really stood out to us, this 1B and 1C, which lowered the MIC of polymyxin 16 to 8 mg per ml. And this was really promising because this told us that these compounds were enhancing the antibiotic activity. And more importantly, this allowed me to determine that compounds 1B and 1C were probably going to be the best compounds to move forward for probe development. And this is exactly where we headed from here. So once more, using that substitution reaction of an aryl bromide and then TMS deprotection, I synthesized P1 and P2, so probe 1 and probe 2, in a 75 and a 46% yield. Leveraging that activity assay once again, I wanted to assess their potency and if they could bind to the catalytic domain. And yeah, they could. However, the addition of this alkyne did perturb the binding to the catalytic domain as well as the overall potency of these analogs. So much so that this P2 analog was essentially inactive. So despite these alkyne-mediated effects, I wanted to push forward and start some preliminary pull-down assays with these guys. And for those of us who are not familiar with what a pull-down assay is, we'll take a look. So this will typically start in something like a whole cell. We can then treat this bacteria with our desired probe and allow it to label any of the target proteins. From here, I can use that click reaction to click on a biotin group, utilize streptavidin functionalized magnetic beads to physically pull these proteins out of solution, and finally release them to conduct our traditional proteomic analysis to get the protein ID. So the next few slides are going to focus on the optimization of these three steps. In this first experiment, the goal was to determine if I could actually click something on that alkyne using an azide alkyne copper-mediated cycle addition. Specifically, I wanted to know if I could click on a tamrafluorophore. And also, I wanted to determine if I could label that model HK53 protein. So for those of us who may not be familiar with looking at gel data, we can simply think of gels as TLCs for biological samples. Since I know the molecular weight of my target protein, I can utilize this ladder to correlate the signals in the gel with the actual protein identity. So in the untreated lane, we wouldn't expect any fluorescence emission. However, in samples that were treated with that probe, we would expect some type of TAMRA signal, specifically to detect our model protein. And that's exactly what I saw. Walking through this really briefly, untreated, no signal, great. P1 treated samples, signal correlating to 32 kilodaltons, and some additional proteins. So this was great because it told me that my probe was PUT compatible, and it could also label that model protein. So from here, I needed to figure out if the labeled protein could be enriched and released using those magnetic beads. So once I incubated these samples with the magnetic beads, I remove any unbound proteins. From here, any remaining proteins in solution with the magnetic beads, I go ahead and release them by treatment with 2% aqueous hydrogen. So the same signals that were detected in that initial experiment would be exactly what I would expect to be in after the cleavage of these proteins. And that's kind of what I observed, where in the untreated lane, there's no signal, and in the lanes that were treated with the 2% aqueous hydrazine, we are seeing that band somewhere around the 32 kilodalton region, indicating that low cleavage of this HK53 protein had occurred. So I trialed various optimizations, and I'm only going to be able to show you one due to the lack of time. So I wanted to trial different percentages of this aqueous hydrazine to hopefully boost this cleavage higher. So in this next experiment, what I did was I took the unbound proteins, and I was specifically looking for the reduction of signal correlating to this HK53 band. And that's what I saw. In the untreated lanes, we still have that presence of the HK53 band here. However, in those that were treated with the probe and also the magnetic beads, we can kind of see a reduction in that protein band. So this was telling me that the enrichment was occurring. So moving on from here, I went ahead and I trialed two concentrations of the hydrazine, a 2% and a 5%. And unfortunately, I was unable to detect any HK53 post-cleavage. So this was telling me that P1 was unsuccessful at enriching and releasing the target protein. And more optimization would need to be required before this methodology could be applied to identify the cellular targets of our inhibitors. So just to summarize some of the work that I've done on this project, I went ahead and I generated a series of covalent inhibitors. I assessed and characterized their activity using an in vitro protein assay, in vivo viability assays, as well as a virulence assay. And 2 of the 4 analogs that I synthesized were promising candidates for probe development. And unfortunately, despite all of this effort that I had put in previously, these probes were essentially inactive in essentially pulling down our model protein. But some of the broader implications of this work was that I was able to show that covalent inhibition of these HKs is a promising avenue that we need to investigate further. And by doing so, we can deepen our understanding of these proteins as well as the generation of antivirulence therapeutics in order to ensure the health of future generations to come. And before I, stop talking your ear off, I was really, dissatisfied with the progress of this project, specifically with the probe reactivity. It really sucked to have put in all of this time and effort and just not get the result that I wanted. So in order to continue trying to answer this question of the cellular IDs, I'm now developing an alternative methodology that's known as a cellular thermoshift assay. This allows me to detect that protein ligand binding by changes in the melting temperature. And it allows me to bypass the need for any compound modification, and it is amenable to high throughput screening. And with that, I just want to say thank you to everyone who was attending my talk and everyone who spent so much time, setting up this symposium. It's really nice to see everyone's work over the past 3 years. And with that, I will take any questions.